Third. I should warn you, I'm drinking coffee. So this is about an hour-long talk, so the more coffee I drink, this might turn into a 20-minute talk. I do tend to get a big side. So Andrew's not here today, so I get to introduce myself. For one, just thanks to everyone for coming out today. Usually Sunday afternoons, I don't know about you, but Sunday afternoons for me, that's nap time. Right? So thank you for sacrificing your Sunday afternoon nap time to listen to me talk about stuff. So some of you did catch a previous talk that I gave was about mathematics. And mathematics and reality, they seem to integrate into a very interesting presentation on what God has done with the universe. Today I'm not talking about mathematics so much. So some of you may breathe a sigh of internal relief, right? Thanks for that. But so my background, I didn't really talk about my background too much. So if you don't know, so I'm a mathematical biologist. I've studied intracellular calcium signaling. So inside our cells, calcium is used as a trigger for activating muscle contractions, neuronal firings, and all kinds of a variety of things. So over the years, as I've done studies in mathematical biology, I've encountered some interesting things in biological stuff. Because of course, with a background in mathematics, if I want to apply it to biology, I actually have to study biology and know what the heck the biological systems are before I start applying mathematics to it. So the stuff I will talk about today are some of these things that I've discovered that made me go, Have you ever encountered things that make you go, Hey, Leon. How's it going, eh? So just a few of the things over the years that I've encountered that made me stop and go, that's interesting, especially from a Christian's perspective. So why the heck do we have 7 days in a week? What's the deal with 7-day weeks? Is it just it's just a historical convenience? Is something that was imposed on us by some king at some point, just like the imperial foot? We don't use feet here, do we? No. What are these things for if we don't use feet? Oh, to walk. Okay, that's it. Also talk about a branchless tree of life. We've often been told about evolutionary patterns and say that all things have evolved from a single proto-organism way back in the day, and that everything's connected through a tree of life. Also talk about a mother of us all, that there's some interesting evidence that suggests that there's a single mum that we all come from. And finally, would anyone care for some immortality? It depends, right? Like, how do you mean immortality? Okay, so first of all, let's jump into the 7-day week thing. So why 7 days? So if you notice on this, the 7-day week is firmly entrenched and no one knows for certain where it came from. Perhaps the week was originally used to mark the phases of the moon, and we have forgotten the connection by a fellow Brown Man in the Stars from 1978. Our own Western 7-day week, one of the most arbitrary of our institutions, came into being from popular need and spontaneous agreement, not from a law or the order of any government. The number 7 almost everywhere has had a special charm. The Japanese found 7 gods of happiness. Rome was set on 7 hills. The ancients counted 7 wonders of the world, and medieval Christians enumerated 7 deadly sins. This is from Borson and Borson and the Discoverers in 1983. In what ways is the 7-day week an optimum time interval for our physical and mental well-being? Don B. DeYoung, PhD, the 7-day week. Maybe there's some historical views we can take to get a little better idea of what's going on here. So the Jewish, of course, based on the 7 days of creation. Is that familiar to anyone in here? Okay, just checking. So the Babylonians, they had significant days on multiples of 7, 14, 21, 28, 49. The Persians, Cyrus the Great, mentioned 7-day weeks in the 6th century BC. Hindus, some speculated they had 7-day weeks approximately 1700 BC. The Chinese originally used a 10-day week, then appeared to use 7-day week by the 4th century AD, and the Japanese followed suit by about 1000 AD. So the Mayans, they had apparently 13 20-day weeks, probably in concert with, they liked base 20, I believe. Has anyone studied or looked at Mayan mathematics? So they worked with base 20 stuff. Whereas like the Babylonians, they liked base 60. So that's why we have the 60 seconds and 60 minutes and an hour and stuff, because they fooled around with base 60 mathematics. So fortunately for us, we have base 10. Right. So historically, there are different kinds of weeks, and people have used different numbers for these weeks, and it is varied. But the 7-day week origin is not entirely clear. Digging into the history of the 7-day week is a complicated affair. Authorities have very different opinions about the history of the week, and they frequently present their speculations as if they were indisputable facts. In short, nothing can be said with certainty about the origins of the 7-day week thing. Isn't that curious? Well, of course, most of us here are reasonably familiar with the Bible. So there's some mention of 7-day weeks in the Bible at some point, right? So look at some of the religious origins in Genesis. By the seventh day, God had finished the work he'd been doing. So on the seventh day, he rested from all his work. God blessed the seventh day and made it holy, because he rested from all the work of creation that he did. And of course, these religious origins would be an irritant to some individuals, entire societies. Excuse me, atheist societies have tried to erase the 7-day week altogether. And just a couple examples I have here. Is anyone familiar with the French Revolution? Of course, the Soviet Union and their approaches to change society. So the French, their revolution, they actually went to extraordinary lengths to redesign a society, attempting to establish a humanist republic. They included the ideas of the so-called Enlightenment, and they glorified human reason. They actually built temples to human reason, where you could go in and you could worship reason. Okay. Right. They specifically attacked the church, right? So the Catholic Church, much anger and resentment against the Catholic Church. Some of it wasn't unfounded. The Catholic Church did do a few things with property and whatnot back in the day. So they had some animus toward it, but they took it a bit far. They invented what's called a decimal calendar. The year was divided into 12 months of 30 days each, some extra days here and there apparently. Each month was marked by 3 decades of 10 days, and on the 10th day you got a day off, so it was like basically a 9-day work week. And they changed the days into 10-hour days, because they were really keen on the decimal thing, to get rid of the Babylonian base 60 stuff. So 10 hours of 100 decimal minutes on 100 decimal seconds, and so for them an hour was, to us, about 144 minutes, it was a little over 2 hours, and a minute was like roughly about what we would have with a minute and a half, and the seconds were kind of short. So they were just trying to redefine all this stuff. And it lasted, about 12 years before it basically fell apart. It didn't really survive too long. The Soviet Union, they were working with 5-day weeks, 4 days of work, 1 day off, so maybe that sounds appealing. Right, so you work for 4 days and then get your next day off. The schedules that they did, they were staggered. So they had these color-coordinated scheduling systems. So the factories were constantly running all the time. I always want to say when I mention this, but they were running 24/7. Right, but that doesn't really apply. Right, so 24/5 or 24/4. Yeah, so people were always working, and the factories were constantly going. But unfortunately, it caused problems with some people. Part of in a family, they couldn't be together. Fathers and mothers, children. They might have totally different days off. So it fragmented society, and some have speculated that was the whole purpose. That so Marx and Marxism, they weren't that keen on the family unit, on the nuclear family. They were keen on redefining society at a fundamental level. So society was basically divided into 5 different groups, all defined by what days off they had, right? So friends, social groups, families were all forced apart or together, all based on what day of the week they had off. And of course, eventually, the 5-day work week was removed. They went to a 6-day week, and then everyone had the same day off. And apparently there's a very large social pressure in order to get so that everyone could enjoy the same day off, right? Of course, the motivation behind a lot of this, as with the French, was to erase any vestiges of religion or vestiges of some sort of created order suggested by what's in the Bible, right? Okay, so that's the political angle. So what do I say there? Oh, so yeah, and only Saturdays and Sundays were eliminated, the days with Sabbath significance. Okay, so that's the politics. So I didn't really want to talk to you about politics. Thank goodness for you, right? So we talk about biological stuff. So I'm sure you've heard of rhythms, that we have biological rhythms, intrinsic rhythms, right? We have rhythms that extend from different scales. So we have circadian daily rhythms, right? So how many of you have not heard of circadian rhythms? So everyone's familiar with that. So are you aware you have daily rhythms? Do you wake up and fall asleep every day? Right, so it's a very familiar thing, right? So circadian rhythms. We also have very fast time scales. So our guts are generating contractions every 3 to 10 seconds, depending on which bit you're in. So that's what I did my PhD on, was studying what are the mechanisms to generate these contractions. So you have very fast time scales. You also have longer time scales, monthly at menstruation and so forth, and some longer, it's called so-called infradian scales that are longer time spans. And so, of course, there's data supporting some of this stuff. So over the course of a day, you have all these different aspects, biological, molecular aspects, cell counts, your blood pressures, your respiratory rates, and whatnot. They vary over the course of a day. So that's fairly familiar. But so how many of you heard of weekly rhythms? So we actually have these things called circaseptan rhythms. And so circaseptan literally means a cycle consisting of 7 days in which biological processes of life resolve. So that's basically the Wikipedia entry. And so I don't know about you, but sometimes I go to Wikipedia, it's like, what do I do with this mathematical thing? Okay, thank you to get a reference here. And so I look at some biological thing, and it's, oh, what is this? Oh, yeah, it's a runoff. And then it's a good starting point, but don't land there for all your information because you find something like this, right? And the last time that I looked at it, there's a reference on it. It doesn't talk about what are these processes and so forth, but so the last time I looked at it, it does include a reference to an evolutionary paper, presumably to suggest an evolutionary reason for 7-day patterns in biological systems. And it just puts it in there as a reference, and it's not tied to any of the text for what it is. Okay, so what the heck are these things? What are these 7-day patterns? So this is taken from a paper from back in 1991, and some of the data, of course, is noisy. It's a bit shifted off, not always precisely on the 7 days, but there are 7-day patterns that emerge for various recuperations. So say if someone has jaw surgery, you tend to get swellings that follow 7-day patterns. If you have scarlet fever, your body temperatures tend to follow 7-day patterns. After bloodletting, you have cell counts that tend to follow 7-day patterns. Same thing so if you have a kidney removed, you tend to have 7-day patterns. So what the heck's going on with that? Is that just old data that was from 1991? So for any of you that studies biological stuff, if you read a paper from 1991, that's ancient history, right? So this one's a little more recent, 2016. So this one's more for tracking kidney rejections. And so completely different locations in the world. So there happens to be these spikes that all correspond to 7 days and multiples of 7. Interesting. So is there a medical interest for this? From the medical point of view, so-called circaseptan, about 7-day reactive periods are of predominant interest. This periodicity can be observed in numerous adaptive and compensating processes. It does not depend on the external week cycle and was already known to the antiquity. So when I bumped into this stuff, I know a few GPs here and there. So I asked them, so what do you think of this? And you know what their first response was? It's just the calendar. Right? It's just because everyone's running on a 7-day cycle. So it's just imposed externally. Which there's some validity to that argument because there tends to be more heart attacks on Monday than on Saturday or Sunday. I wonder why. Right? Probably math tests have something to do with it. But then the other response was more, it's like, oh, how do we schedule stuff like that? It's hard enough to book someone for a surgery, much less fool around with their 7-day patterns. So go talk to the administrators and the bookkeepers and whatnot and try to get them arranged around a 7-day pattern. Forget it. We're lucky enough just to get them in there in the queue to get their surgeries and stuff. And so that was just an interesting response. It's like, that's just too complicated to deal with. All the same, though, this has been known for quite some time. Hippocrates and other ancient physicians observed these 7-day cycles emerge in people recovering or suffering from disease. So this has been known for quite some time. Isn't that interesting? Somehow this knowledge is rather obscured. I'm going to go to the Wikipedia page. Doesn't really describe at all what this thing is about. How many people here actually ever even heard of circaseptan? Right? It's just circadian. You hear all about circadian. Circaseptan is like this totally unknown thing, and I bumped into it a while back. And actually, I was keen to write a review paper on the thing, and then someone beat me to it because it's like, we should be studying this. No one is studying it. No one seems to care. And there's just a few papers that have come out. I was like, hey, we should pay attention to this thing. So, in other words, I guess the whole point of all this is have a day off. Right? Be sure to rest and recover, right? This 7-day period is for reconvalescence and healing, and this is coming from a biological perspective. I didn't know if that was commentary on what I was saying. The 7-day week is, of course, embedded in a world through societal constructs and so forth. They synchronize us with each other, so we're going to have the same days off, but we also have intrinsic within us 7-day rhythms that are intrinsically related to recovery and convalescence. And so when I saw this, of course, it made me go, Remember the Sabbath day by keeping it holy. 6 days you shall labor, do all your work, but on the seventh day, kick back. Right? I think that's the Shawn version, the Shawn version of the Bible. Right? So, any implications from that? Right? Okay, so that's circa septans. So that's one of the 4 things I wanted to talk about. So the other bit is, of course, this tree of life, and how evolutionary theory posits that all life is related. Typically, we see lots of diagrams like this. We take freshman biology or something. And you hear about these leaves, these individual species. They're all related to each other, all to a protoblob that started life way back when. So let's say one of the details for this is that you have to find evidence for it. So it's very nice to have theories in science and whatnot, but it's good to have evidence that supports your theory. And so, of course, the original idea was to, well, let's go digging in the rocks and see if we can find fossils that support this, that fit all these species together. So paleontology is the study of plant and animal fossils, and some big shot paleontologists back in 1973, they proposed a theory to explain the lack of fossils that were filling in these branches, otherwise known as punctuated equilibrium. And so Stephen Gould had this interesting thing to say: the extreme rarity of transitional forms in the fossil record persists as the trade secret of paleontology, the trade secret. Like they weren't telling anyone. The evolutionary trees that adorn our textbooks have data only at the tips and nodes of the branches. The rest is inference. So it's like, so these bits that connect everything together, that's not coming from fossil evidence. You might be able to fill in a few bits and infer a few that suggest things. But so science doesn't stand still. There's always more grants to apply for and more PhD students to crank out. So science moved on. So it looks beyond the bones, right? So there's other things that we can look at. So proteins, you are made of protein. Did you not know that? Right, so your hair is made of protein, right? So hair is made of protein. Your flesh is made of protein. Your fingers are made of protein. So also this bug is made of protein. So also that plant eating bugs is made of protein. So we have protein structures eating protein. Protein, right? So protein are fundamental units that are built, used for building all organisms, and they themselves are made up of amino acids. Has anyone heard this stuff before? Right, so here's just a quick overview of some biological things. So basically proteins are made up of these Lego units that are all stacked together, and there's actually a kid somewhere that is just making proteins out of Legos in a back room somewhere, and he's cranking them out. Oh, look, here's your hairstyle. Right, but so molecular biology is the study of biological molecules such as these proteins, and in particular, of interest now are these amino acid sequences. So these individual sequences of these Legos, right, so you can take individual amino acids, and the way you chain them together, you can have dramatically different properties for individual proteins. So even though they might functionally perform the same tasks, they might be assembled in a very different way. Right? So these assemblies are all built on about 21 different amino acids, or 21 different Legos, right? And so the sequence or collection of these, they provide a way to study relationships between organisms, or similarities or dissimilarities, right? And so what you can do is you can say, oh, here's a common protein that's in people and mice and rats. And then you can look and see what's the sequence of these amino acid building blocks, compare them and see how similar or different they are, and try to make an inference about their distinctiveness, their relatedness, or depending on how you look at it. So people did this. In 1973, these people, Dayhoff and McLaughlin, compared sequences of amino acids in a protein found in all animals. And this protein had to do with production of energy in life forms, otherwise known as cytochrome C. Does anyone know what that is? It's important. I see one nod, yes, okay. So this is important. Without cytochrome C, we wouldn't really be doing anything. It would just be blobs of protein. It helps us to process energy and keep doing things. Right, so when they compared all these sequences across a vast number of different organisms, like mice and clams and fish and people and monkeys and whatnot, and they said, hey, we found an evolutionary pattern. It supports evolution and the whole tree of life idea. And so perhaps that they sidestepped that issue with the trade secret of paleontology, that the fossil record doesn't support this stuff. So maybe we found the support in molecular biology. So hooray, a tree of life. Fast forward to 1985. So has anyone heard of this fellow, Michael Denton? He wrote this book back in the 80s called Evolution, a Theory in Crisis. He's also written a sequel to it recently, Evolution, Still a Theory in Crisis. He was formerly at Otago University. Now I think he's with the Discovery Institute. Steve Meyer and his gang doing intelligent design. But so there's another perfectly valid interpretation of these protein sequences. Instead of assuming that it means degrees of relatedness, it's just difference, just how distinct they are from each other. Denton, in this book, he analyzed the exact same data set that Dayhoff did in their 1973 paper. And so with just a different presumption about the data, he found an alternate and still yet statistically quite valid pattern reminiscent of a 19th century view of biology before Darwinian mechanisms became dominant, called topology. So let's say is that 2 bits today? So we had circumception. So I introduced you to circumception. So that's your favorite word of the week. You're going to be talking to people about circumception. Have you heard about so how about, is this the second favorite word of the week? Is it typology? Typology. What the heck is typology, right? So what is typology? It has to do with types, right? It's variations on a theme kind of thing. Topology classifies organisms into types which are basically like equidistant from other types. Within each type, there are variations from like an archetype that you may or may not actually see. So let's take an example. So maybe I lied a bit to you. There's geometry. So it's not really mathematics, right? So you're getting geometry. So does anyone recognize this little fellow? What's he? Square. He's a square. And so this little fellow, what's he? Triangle. He's a triangle, right? And so I've put equidistant from these. These are all triangles, right? So the square is fundamentally different from triangles in very key aspects. So this fellow has 4 sides, 4 angles, and these fellows have 3. And you flip it around, so he has 3, and these have 4, right? So that's one way of saying these are square types, quadrilateral types, and these are triangle types. And so we can also organize these families of quadrilateral types. So no matter how much you vary these things around, these are still quadrilaterals. No matter how much you vary these things around, these are still triangles, right? So we have an archetype, and we have the variations on those types. And so topology basically applied that concept to biology, right? So we have an archetypical cat. Does anyone recognize these 3? So I think he's Felix, right? So that's Eek, and who's this? Stimpy. Thanks. Yeah. My math students at Massey, they didn't know Ren and Stimpy, and I was shocked. It's not cultured. Right, so we have bugs, right? So this one particularly is an insect, right? 6 legs, 3 body parts, stuff like that. So you can have types of these that also further can go into larger and larger groups. Cats are types of mammals, mammals are types of vertebrates, and so forth. And so what's interesting is how these types, given this typological view, would be equidistant from very different creatures. So this suggests that cats are just as different from bacteria as they are from birds. That insects are just as different from cats as they are from reptiles. So to many modern minds that have been trained by looking at things through an evolutionary lens, this sounds bizarre, right? And it so happens that Denton, when he looked at this data, that's pretty much precisely the pattern that he found. Right, so he found equidistance for gastropod mollusks. It's basically, is it an oyster? Right, oyster, clam? Oysters and clams are not the same thing, are they? Are those just things that you eat that make you ill? Because I know someone gave me some smoked clams or whatever, and I was very unhappy. But so apparently the smoked clams, regardless of how you cook them, they're equidistant from such disparate things as lampreys, carps, fish, frogs, amphibians, chickens, birds, kangaroos. Kangaroo, it's not a mammal. What is it?
Marsupial.
Marsupial. Oh, thank you. So these fellows are equidistant from such totally different creatures. And this is the pattern that Denton found. And these typological patterns just extended throughout, and he made this conclusion. In terms of their biochemistry, none of the species deemed intermediate ancestral primitive by generations of evolutionary biologists and alluded to as evidence of sequence in nature shows any sign of their supposed intermediary status. So he approached the exact same data and had a different presumption, and he got a very different conclusion. A very different conclusion, right? So he put this all together, and what's that saying? It's saying that these presumed connections between major groups of organisms is not supported by that assertion. What a different perspective, right? Again, like I say, this is typically jarring to contemporary minds. This looks bizarre. And I would say that that's because of an observation that was made by a fellow, Dwayne Gish, that wrote into the American biology teacher in 1970. How many of you existed in 1970? Okay. I feel much better in this crowd. 'Cause, like when I did this at, that home group, right, Andy? Right, went to the home group. How many of you existed in 1989? And like, Andy and I. Right, so thank you. Right, so this is what this fellow had to say in 1970. The dogmatic fashion in which evolution is usually taught in our schools and universities amounts to indoctrination. And it is as much the teaching of religion as if the theory of origins restricted to the book of Genesis. So he's suggesting that way back in 1970 that you have people being indoctrinated with only one way of seeing things. There are actually different ways of looking at the exact same data and arriving at completely different results. This conclusion, of course, by Denton has been widely disputed. This is from 1985, so don't think for a moment there wasn't a response. Right? Saying that Denton doesn't understand the data, he doesn't understand phylogeny, these phylogenetic trees. These distances only appear equal because they're from different nodes on the branches and so forth. That's what he's actually saying. But if you can actually go get the data for yourself and just look at it, that's what I did. It's very easy to obtain. And just pop it into Microsoft Excel, and boom, boom, and you see a totally different alternative interpretation. And so again, that's something that made me go, as a mathematical biologist, that I happen to be a Christian, right? A typological view of biology merges with this totally different initial assumption. And it seems to resonate with this thing about kinds, right? So in Genesis, there's this talk about God created great creatures of the sea and everything which with the water teems and moves about according to their kinds, every winged bird according to its kind. It's all about kinds, right? So that's two. Does anyone just like saying Okay, so that's it for today. Let's get out of here. Let's go have a nap, have our Sunday afternoon, and play video games once we wake up. Oh, that's not what I do. Mostly. Right? Oh, no, actually, I want to say thanks to Mum. I'm glad Karen's here today, because she's a mum. See, she brought Indy, right? Thanks, Mum. Right, where would we be without mums? Well, we wouldn't. Right. We wouldn't exist. Right. So of course, this is kind of a requirement whenever I do a talk, is you typically see some Simpsons thing, right? Because from the '90s, I was drilled into the head with the Simpsons, right? So if it wasn't for Eve, where would we be? So once again, we talked about what amino acids and stuff, and the building blocks for proteins. Well, it turns out that these proteins themselves, right? So the proteins that make us up and their building blocks of amino acids, there's actually something behind all this. That's sort of a blueprint for these things. Is anyone familiar what these are coded by? What's the magic three-letter word I'm looking for, huh? DNA? Do I hear AMP or ATP or STP? No, do you want to go with DNA? Are you sure? Because I think you're right. Yeah. DNA, right? So DNA, what is DNA? It's this double helix thing. It's a bunch of molecules that are woven together, right? And so this is sort of a blueprint. So like the individual base pairs, the molecules, they actually map over directly to the amino acids. Which kind of amino acids, right? So DNA itself can be packed into chromosomes. These chromosomes can be packed into nucleuses. The nucleuses are all part of cells. And so just maybe you find this disturbing information, but you are full of cells. A lot of cells, right? And these cells are themselves full of stuff. Cytoplasmic, mitochondria, nucleus, plasma membrane. And so every time I give a talk, and this word is in here, I always ask my audience to join me in the joys of saying it. You join me? Ready? Endoplasmic reticulum. Thank you. I just think it's the neatest word, endoplasmic reticulum. Right? Yeah, neat. Okay, thank you. And I do that at technical conferences. So I have a bunch of mathematicians saying endoplasmic reticulum. Okay, so the bit that I'm actually mostly interested in is this mitochondria thing. So mitochondrion, let's get to that. So mitochondrion, what is the mitochondrion or mitochondria in general? They're the power generators, right? So these are the guys that are handling the energy needs of the cell, right? They convert glucose into the gasoline that the cells need. It's called adenosine triphosphate. So it's just basically a molecule that it can snap off. The cells can snap off one bit. There's a bunch of energy that it can use for driving all kinds of processes, doing the things that cells do. Play video games, take Sunday afternoon naps and stuff like that. So of course, the day-off data I showed you was for a cytochrome C that sits in this mitochondrion. See, I think I got another picture of, yeah, here we go. So mitochondria themselves, it just seems like the deeper you dig into the worlds, it just keeps going, right? The complexity just unfolds. So cells are quite complex. You dig into individual cells, and then you have parts of the cells, and these are quite complex. Inside these things, mitochondria actually have DNA of their own. So there isn't just the DNA in the nucleus. There's also mitochondrial DNA, right? So and that's for the small bits of proteins that they need to make for their own functions. So let's say they only come from mom. Mitochondrial DNA, all the mitochondria in your body and all the DNA in the mitochondria are from your mother, right? So they're only passed through maternally. So the contributions from dad, they all go into the other DNA bits. The stuff from mom is all tracked through the mitochondria, right? So it provides a way to look at histories of people. Who's your mom? Who's your mom's mom? Who's a bunch of people's moms, right? So they're useful in that sense, right? So partly because of the normal nuclear DNA that codes for like all the proteins that make up your hair and your fingernails and your tongue and your eyeballs, yeah, it's complicated, right? So that set of DNA is like 3 billion base pairs, so it's massive. And there's only small numbers of copies. Oh, here they come. Should we lock the door? Get invaded. Little kids wearing eye patches and stuff. But so you may have 2 copies of these things, and so that's a lot of base pairs to work with and not many copies. Whereas like mitochondria is pretty short. It's only about 17,000 pairs. And you get hundreds of copies in an individual cell, so you have more of this DNA that you can work with, right? So, and it provides another way to compare relatedness, say, of organisms. So who's your mom, right? So you can track through, say it only comes from the mother, so it allows you to track maternal lineages, right? In particular for humans and so forth. And so as taken from a textbook, contemporary patterns of genetic variation, particularly in genes carried on mitochondria, provide information about the origin of human beings. How profound. This is from a textbook that I bumped into in my work at the university, Boyd and Selk, How Humans Evolved. And so we're going to be using them to explore this a bit. So of course, mitochondrial DNA provides a window into these things. And what's interesting is that the human species is less genetically variable than other species when looking at it through mitochondrial DNA, which is a bit surprising because I'm just looking out at you people and you're pretty varied, right? There's quite a bit of variety there. But so the number of differences per DNA base in mitochondrial DNA across the geographic human populations, Africa, Asia, and Europe, is really very small. The number of substitutions, the number of changes in a nucleotide is fractional. It's so small. So it's kind of hard to see that without a comparison. So by comparison, let's look at this one. So this is for chimpanzees. So the smallest distance here for chimpanzees is the largest difference for people. Right, so that just, when I first saw this, I was like, really? That's weird. Right, so what does that say? So people aren't as different as we think, or at least different in the sense of who their mums are, who our mums are. Right, so what does that mean? And they calculated, given that an assumed rate of mitochondrial DNA mutations, the way they change, looking at the current amount of variations for people for these mitochondrial DNA, the calculations that came out said there should be 5,000 people in the world. Oh, 5,000 women, sorry. It's maybe 10,000 people, give or take. Is that right? There are 5,000 women in the world? How many women are in the North Shore? right? So there's something wrong, right? So that estimate is wrong, right? So there were suggestions that there's something else happened. There was these tiny variations suggested some sort of population explosion. The most likely explanation for the small amount of genetic variation observed within the human species is that we've undergone some sort of population explosion. Right? So there was a rapid spread of people across the face of the earth, right? And so let's see, I think so this chart, the fact that modern African population, this African variation, right? So this one was the greatest one by far, right? Even though itself is rather minimal. The fact that modern African populations are genetically more variable than others is consistent with the idea that the population explosion started in Africa. So that's where the human population started and then spread from there, right? Because it also had more time to get varied. So let's see. Yeah, there's a small group of humans diversified and changed into the races we see today. So I think this is also known as the out of Africa hypothesis, or the Garden of Eden hypothesis. So somewhere in Africa, maybe there was a Garden of Eden thing, I don't know. I haven't actually been to Africa, so this contour plot is just showing you how the variations were spread out spatially, geographically over this. And so of course the highest variations are here in Africa, and it just suggests that there was a spread, right, with some alterations here and there. Maybe this is the Spanish. The British came along when they visited North America. The fact that mitochondria are inherited maternally means that the mitochondria of every living person are copies. Let's see, is this up here? Are copies of the mitochondrion of a single woman who lived in ages past. That's intriguing, right? And so they gave her a name, also known as Mom. Thanks, Mom. Right. Also known as mitochondrial Eve. I think it got popularized that way. There's mitochondrial Eve, like the Eve who started this whole thing. Right. This incredible idea is the simple consequence of asexual inheritance. Right? It's just mitochondria just comes from mom. The logic is based on the fact that in each generation, some women just leave no female descendants. So it's like there's a certain percentage, right? So Karen has Indy, who is a girl. So yeah, your mitochondrial DNA continues, right. I was like, oh yeah, thank goodness. I was like I'm an only kid. Maybe that explains things. right. So I'm an only kid, so mom just had a boy, right? So it's just like I'm not, her mitochondrial DNA is not going to continue for me. So it does complicate things, but it also makes it easier to track some of this stuff and to see. Eventually, everyone carries the mitochondrion of a single woman, the most recent common ancestor, who's sometimes given the colorful but misleading name Eve. So some of the researchers are really uncomfortable with that name because of the implications. Mitochondria, oh crud, you're just going to have all these creationists jumping up and down. Right? So I want to get into some of the statements that tells me Eve was an otherwise unremarkable member of a large population. So they basically say Eve wasn't a single female. It was actually a group of females that had all the same mitochondrial DNA. So that's one of the statements they make. But the population whose only claim to fame is that her mitochondria happened to survive while the mitochondria of other women did not. So they tried to explain this, and this is going through this textbook, right? So tell me why. Why do you think this happened? So in each generation, some women leave no female descendants. So this diagram just makes me think of Plinko. You drop a coin, tick, 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 right? So here we're dropping mitochondrial DNA, tick, 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 tick. Right, so Eve here, apparently her mitochondrial DNA spreads, and it managed to survive because these just track the number of daughter offspring that she has. So say this person didn't have any daughters. So the mitochondrial DNA line, done. This one, it made it so far, and then it stopped. This one did pretty good, but it stopped here. And so they're saying that this graphic just illustrates how one mom, her mitochondrial DNA can just take over just because she survives, and her offspring survive. But so they say this mitochondrial DNA tree for a hypothetical population of 12 women shows why all the mitochondria in any population are descended from a single woman's mitochondria sometime in the past. In each generation, some women just leave no female descendants. So that seems reasonable enough. But if you get into just the specific wording, there's a chance that one mitochondrial DNA lineage will be lost because all the descendants of one of the women will have no daughters. Eventually, the empty DNA of all but one will disappear. So you're saying, so this graphic is supposed to illustrate, where does it say it? It's exactly the same thing will happen in all populations. That's an extraordinary statement. All of them, everywhere, throughout all time and space. Whether you're in a video game, or you're on TV, or you're in Auckland, right? Everywhere. Okay, so I'm a mathematical biologist. I use MATLAB. Does anyone know what MATLAB is? Okay, so let's do some mathematical biology. Yes. Okay. So I lied to you. There is some math. Right? So what I did is I wrote a simulation. So I often make simulations of these things. So let's see, I've got some code here. So just a script for testing out this mitochondrial Eve explanation and Boyd and Silk. So this thing is just a simulation, just to see, okay, so I've got so many mums that I start with. And I say, here I start with 10 mums, and I go for 10 generations. I have the mean number of kids that they have and the standard deviation for the number of kids. So this is saying on average, mom has 5 children, right? And so then, so I create a distribution of these based on these. So many of the kids that come out. And so then I have a probability of having a daughter. So 50%. Does that seem reasonable? 50% chance of having a daughter, right? So I go through and I run just this code, just calculating based on the number of generations, whether or not they survive and who goes extinct and so forth, and I plot all this stuff. So let's see. Let's try instead of what's the average number of kids people have today? Like 2? Let's go with 2. So the standard deviation. So maybe 2. So you have like maybe 4. Some people have 4 kids. Right. So let's try that one. Let's try this. Let's see if it's going to work. I have to change this thing. So this is what I do for my job all day. But it also involves going, no, right? It's not working. Okay. Yeah, so I usually have QWERTY. Okay, so initially this is just the distribution of kids that are born. So what I've done is like the average here is 2, and the standard deviation goes up 2, and there's a second deviation. So you can have 4 kids, or some very few people at 6. But so the rest of them, they would generally come out this way. The mathematics doesn't care if people have negative children. What does that mean? I have negative children. I mean, not children that don't eat their vegetables and refuse to obey, right? So it's just negative. It doesn't make sense. So what I do is I combine them all in so that you just have no kids, right? So that's where the distribution falls. And so what this is just saying, okay, so we've got a certain percentage of survivability, we've got a certain percentage of them are actually going to be girls and so forth. And I start off with 10 mitochondrial lineages. And the number of extinctions here is pretty high. So 7 of these lineages go extinct, but by no means does only 1 survive. Right? And so that's usually like a contemporary sort of number of kids that you have. So is that different in the past? Like in more agriculturally based societies, how many kids did they have? A lot more. I remember my grandmother, she grew up in the Depression in the Panhandle of Texas. Guess how many brothers and sisters she had? Right? Well, let's change it to 13. Right? She had a lot. And that woman could roll cigarettes with one hand. It was amazing. She was just, Grandma, how do you do that? Right? 'Cause I used to roll cigarettes for the whole family. It was all of them. Wow. Okay, so let's run this simulation. Let's see what we get. Oh, that's a bit more than I expected. So this time, that doesn't look right. That's,
Deviation.
did I change the number of mums? Okay, yeah, sorry. So let's keep this the same, and let's change the mean. Thank you for paying attention to my code manglings. So 13, let's change the average of 13. Yeah, I was going to say, that doesn't look right. Let's see, so we run this simulation. It's still running. It's still running. So this is the other dimension of my life. So I'm running a simulation. Let's go have lunch. All right, so I have a feeling this is going to take too long. Stop. Is it going to stop? Okay. Hi. Okay, maybe not 13. Can we try something smaller so my Mac doesn't explode? What do you think's good? Let's go half of that. 7? Okay, 7. Okay. Maybe we could do this. Okay, boom. Yeah. Okay, so here we have the distribution of the number of kids. So the mean's like 5, 6, 7, right? And so out of the 10 mums, you get 1 extinction. And so what I see in general, like we could run this again. But so with the higher number of progenies, the number of extinctions really falls down. And so as you just look at this more and more, the chances of getting a complete extinction except for 1 lineage, just by randomly running these things, is so low. The probability is really quite low. So when I go back to Boyd and Silk, when they said exactly the same thing will happen in all populations, what do you think? No, I disagree. And that's coming from a computational biologist, which doesn't really count for anything. Right? So, oh, you believe your model, huh? That's nice. Good luck getting funding. Okay, but so if our simulation is more valid than Boyd and Silk's presentation, as long as there's enough daughter progeny, resulting in those first few critical generations, right? So if they get established, then it takes off, because you can see those exponential curves that take off, right? So as long as there's enough to get going. And there's no natural thinning to a solo lineage, unless those fertility distributions are really low, so they're just not having enough kids. There are some alternatives for eliminating these lineages, right? So how could you eradicate these and get down to one lineage? They kill each other, right? So what'd you say about my mom? Right, so they go off when their cities are burning and stuff. So alternatively, you have natural disasters, volcanoes, earthquakes, or whatever. It wipes out some of these populations, right? So I would say that the Boyd and Silk proposal is misleading. They claim it applies to all populations, and they may be stretching things a bit because they're talking to freshman students or whatever. So they're not being entirely upfront. So the data suggests that something happened, a bottleneck of some kind, right? Something dramatically reduced the population. Something dramatically reduced the variation, something smaller, and just some genetic processes just sort of randomly selecting from a population, you can dramatically reduce the variations in these populations. And so that made me go, a natural disaster perhaps, that dramatically reduced the population of human beings? A natural disaster struck the globe, imposing a severe bottleneck on population diversity, reflected in mitochondria DNA. I will send rain on the earth for 40 days and 40 nights, and I will wipe from the face of the earth every living creature. Yeah. Right, so let's just recap. So what was the first one? The word of the week was circumceptin. So the one before that was topology. What's the word for this one? Mum. Just go with mum. Does that work? Okay, so mum. Okay, sweet. Okay, what's the last bit? Okay, so remember this fellow, this cell. A cell, that you have a cell, you have many cells doing lots of things. Thank goodness for that. Cells, they keep us alive and do stuff. So remember this fellow, this tangled looking thing, this X thing, that's comprised of DNA. It's called a chromosome. Chromosomes, they pack in a lot of DNA into them. So have you ever like just coiled up a headphone wire, a headphone lead or something? You just coil. So in some ways that's all it's doing. You're just coiling up these things. But the structures for these, like so the original double helix thing, to get to the chromosomes, it gets wrapped around these beads, and then those beads get arranged in a sort of, kind of reminds me of some of my friends that be stitching off on the corner somewhere. It's like, what are you doing? I'm stitching. It helps me relax. So I wonder if cells do this to relax. They're just relaxing. But so in general, DNA is not packed in all into the chromosome stuff, because actually what that is, it's for when it's replicating, when it's dividing, right? Because it's not easy to get to in that form. Chromosomes do efficiently package all the DNA blueprints for life into these structures for cell division. They're generally only visible during mitosis. I don't know if you can read this, but the net result, each DNA molecule has been packaged into a mitotic chromosome that is 10,000 folds shorter than its extended length. So that's pretty darn good, right? So it's like how long is my speaker lead? It goes, it's 10,000 times shorter. That's pretty darn good, right? All right. So it's for real. These are real things, right? So people have taken light microscopy images of these things, some of these chromosome structures. They're fairly filamentary. So it's hard to get an idea of what's the spatial scale for this, right? I don't know. Maybe I'm just lying to you. Maybe this just is some dog's hair or something, right? But so if you look at the spatial scale, so 2 microns. So anyone know what a micron is? Anyone tripped over a micron high bump on the carpet lately? Right? Probably not. Oh, sorry, I went the wrong way. So we have human hair. So here's a human hair. This is roughly the same width. What is that? That says 1 micron, or this is 10 microns. So this is 2 microns. So 2 microns is maybe about 1/5 of that. And these are all these filamentary structures. That's DNA, right? Wrapped up into a chromosome. Very sophisticated packing mechanism. You can actually see these things in action. Let's see. So when chromosomes, they're packaged up, right? So every time a cell needs to divide, it needs to replicate these copies of DNA, so each of the daughter cells has all the information it needs to generate the proteins for hair and fingernails and whatnot. And so this actually happens every time. All these chromosomes, they have to be divided up. So usually they get aligned down a central axis for the cell, and at some point we should see them pull apart. Right? So now it's just like, okay, so now it's like, wah! This is baby one, baby two. Baby A, baby B. I don't know if they actually cry, and there's like a nurse to smack the cell on its bottom, right? That kind of thing. But so it's actually just really cool just to look at, right? So cool imagery, just watching these things separate. But so this is a necessary part of mitotic division, right? So am I in the right spot? Did I go backwards or go forwards? Nope, here we go. Okay, so in order to pull apart those chromosomes, it needs to duplicate the DNA, right? So it starts off with, a cell has all the copies of DNA in its nucleus, and so it has to go through, and it's like, well, I have to make another copy so I could share it with baby A and baby B, right? Or else baby A is going to be very angry at baby B. Have you ever seen siblings fight over who's supposed to do the dishes? So you imagine cells fighting over, that's my DNA. No, that's my DNA. But so there's machinery that does this, right? There's a physical mechanism. Something has to do it, right? It doesn't just magically happen. So, where's that movie? Show me the movie. You gonna work for me? Yeah, so this is a really amazing movie that got off 10. And so we have DNA coming in here. This protein machine, it's splitting the DNA strands, right? It's a woven double helix. So one strand is peeling off, and it's just being replicated right away. So one just peeled off, right? I'm just being replicating this bit, but it still has to do the other strand. The other strand is a little bit more complicated. So you can see this machine is pinching off loops. It copies these loops, right? Gets the loop so far, and then it pinches it off, right? And it continues, okay, off you go, I've copied you a bit, and I have to pinch off another loop, right? So this protein machine, DNA polymerase, right? It does this every single time. It has to, before a cell can replicate, before a cell can divide, it has to go through this whole procedure. But also, it becomes very important that this bit it has to pinch off. It has to keep doing this. Whereas this other strand is very straightforward, right? It doesn't require so much work in order to get there.
Does that slow down?
Oh, I don't know what the time scale is. I'm sure it's slow. It's quite slow. yeah, I don't know. And that's just another day for a cell replicating DNA. That's just what it does, right? You get these pieces of DNA that are split up and have to be replicated. And of course, this is happening likely right now in you. How does that make you feel? These things are happening. What's that? Okay. It used to be believed that this process, even on a Sunday afternoon, this is happening while Shawn's playing video games, that this would just happen forever and ever and ever and ever and ever, right? Every time a cell needed to replicate and make new baby cells, that it would just keep doing this, right? So it's like no matter how long you would wait just to see how many baby cells are coming out of these cells, they would just keep replicating and replicating and replicating. That this process just went on indefinitely, that it never stopped. Well, it turns out that's not true, right? Buzz Lightyear, yay, right? So actually, what happens is after a while they stop. This fellow Leonard Hayflick in 1961, he discovered that cells stop dividing after so many times. Now it's called the Hayflick limit. Maybe someday I'll have something named after me. The Means video game. He worked very hard on inventing it, and it's not got science involved in it at all. The Hayflick limit or Hayflick phenomenon is the number of times a normal human cell population will divide until cell division just stops. Hayflick demonstrated that a population of normal human fetal cells in a cell culture will divide between 40 and 60 times, and it just stops. So that has to do with what's called senescence. So that's where the cells, they stop replicating. So when they get damaged, too bad, right? That's it. You're stuck with that cell. So that's partly why you experience aging, right? Because there's no new cells to take over and to keep you from aging. Why? Why? What's the deal? Immortality, anyone? Right? It's the machinery, right? This DNA polymerase, that machine that was illustrated. The way that it has to do this, it complicates things, right? So here's yet another video. Let's see if this one will work. So this cartoon will show how, right? It has to split the strand into two strands, right? So this one strand is very straightforward to replicate, the leading strand. It's this lagging strand that requires a primer in order to get it started. So it gets it started, it replicates a bit. This fellow, he just keeps going. He's very easy peasy to replicate. This one requires a primer. You replicate a little bit. That's the pinching off of the loops. It pinches off a loop. So it's this bit that's complicated. Right? It's nowhere near as straightforward as this bit. And this is the process that just keeps going and going and going for it to replicate the entire strand. So as it gets along the entire DNA strand itself, and it wants to replicate, it has to go through and remove these bits, right? And since it has a beginning to these bits, and in between these that have been replicated, it can just fill in the blanks, right? But then you get stuck with this. What's it called? The telomere. So we have these telomere repeat sequences. So even though they get lost every time it goes through the replication process, right? These sequences, they get chucked. So you lose that, it's gone. And so these telomere sequences are only so long, right? So every time it replicates, you lose a bit, you lose a bit, you lose a bit. So what happens as you keep losing bits? I get into important stuff, right? So these aren't necessarily blueprints, don't code for anything necessary. But as you keep losing them, you start chewing into things that are important. And so that's related to the Hayflick limit, right? So the cell goes, oh, stop. I can't keep doing this because now I'm destroying information that I need, right? That's that movie. Do we want to see it again? No, that's enough. Right. So the beginning and the end, telomeres protect our DNA from this replication damage. Right. Every time DNA is copied, we lose that 3' overhang. So it's just a name for these things. You know, leading edge, lagging edge, and so forth. Eventually, like I said, the telomere just runs out. You just lose your telomeres. Let's see. They don't actually code for anything. They're fixed in length. The telomeres, once they're exhausted from all the replications, you get this Hayflick limit. And so think about this. How long are your telomeres? Right? Let's see. Telomere length is directly related to, guess what? Age. Yeah. Because once you replicate your cell so far and you run out of telomeres, your cells stop replicating, and that has a distinct effect on when you start aging, right? Also just disease, because you start getting exposed to disease because your cells don't replicate, create new ones just to overcome the diseases. Of course, mortality rates, and of course the years of a healthy life. Right. So of course this research has been going on for some time now. This isn't a recent revelation, but so these telomeres are involved and well known to be involved with aging in all sorts of mammals, not just human beings and so forth. So it's kind of staggering in a certain sense that at such a fundamental level within your cells, it's almost like there's a blueprint for this is how long you can live, right? That's it, before you start aging. So of course, does anyone want to lengthen yours? All right, so for a nominal fee, we will try to lengthen your telomeres, right? So, eat these nutrients. We'll sell you some nutrients. Just go, these are good for my telomeres, right? Or you want to find out how long are your telomeres? How long do you think you're going to live, right? So you kind of estimate, well, my telomeres are only so long, and I'm not eating enough of these multivitamins for my telomeres, so I'm probably going to die soon. That's disturbing, right? I'm not sure I'd actually want to know. Some creatures running around in the world, they outlive us. There are immortal organisms, right? We don't know for sure because no one's lived long enough to make sure they're still around, right? So the lowly flatworm. Flatworms are immortal. I mean, unless you step on them, right? Oh man, he was immortal till I stepped on him. Right? Sorry there, immortal flatworm. That's more where that came from. Right? Hydras, these little fellows running around in the oceans and such. Some jellyfish. And bristlecone pines. I thought that was a bit odd. So you have these tortured looking pine trees. I think they're native to North America somewhere. So what's the trick? What do they do? Right? How do they pull this off? So if we have telomeres that run out, do you think maybe there's something that can fix that? Right? So if the cells stop replicating when you run out of telomeres, what if you put some back? Would that solve that problem, right? So there actually is a machine called a telomerase. That's precisely what it does. Let's see, where's my cursor? Give me my cursor. Oh, it's running, okay. So this machine, what it does is after replication, it comes along and it goes, I've lost some of this telomere. So it just uses a simple template for the telomere sequence and it just puts it back. So it's, I don't know, it's not rocket science, it's just molecular biology. Right? You just add in this stuff. And so you don't lose these telomere sequences, all because this protein just goes through, oh, we finished replicating, we're running out of telomeres, can you go fix that thing, right? Okay, Charlie, let her rip. Fix the telomeres, okay, you got it, right? That's all there is to that. And incidentally, we have the trick inside of us. It's not a problem with some of our cells, right? So germline cells, like sperms and eggs, they're basically immortal, right? They have telomerases that are active in them. Also, some skin cells to a degree, depends on the skin. I haven't looked too deep into that. But also cancer cells. So that's one of the problems with cancer cells, is something triggers their telomerases and turns them on. And so they abandon ship, they no longer do their tasks in the body, but they keep taking all these resources and they don't die. They just keep going, right? They just keep repairing the telomere sequences. And that's partly why cancer is such a bad thing, right? Okay, so this was, of course, one of those things that made me go, isn't that interesting? Right, so the Bible talked about people living a lot longer than us, right? And so many people have said, oh, that's ridiculous. No one could live, no one's lived past roughly 120 years or something like that, right? And even some ancient Egyptians, they only lived up to about 100 years or so. We nevertheless have the molecular machinery to be what? To be immortal, right? But with a caveat, don't stop eating, and don't step out in front of buses, right? And you can live forever. Although I'm not sure I'd want to the way the world is today, right? So because like for instance, what would be your quality of life? So first this list of Adam lived 130 years. He had a child named Seth. Seth lived 800 years. Had other sons and daughters. Altogether, Adam lived a total of 930 years. And sometimes I wonder, what was that like living for 930 years? Did he get bored? Right. Well, what do you do? Tennis, anyone? I've played chess, I've done bowling, I've done all the sports. Will anyone play tennis with me, please? Right? Jean Calment, she lived to 122, I think. Wow, or 100, was it 120? I thought it was 122. Let's see. Oh, I didn't put it in my notes. Sorry, I think it's 122. But I'm also thinking of my adoptive Kiwi grandmother. So when I first moved here, some of you know my story. When I moved here, I was adopted by an elderly Kiwi, and I lived with her. She gave me a cheap place to stay when I was a dirt poor PhD student. She was a widow, so I took care of her. And she was also deaf, and I'm a drummer, so we got along sweet. Right? But so she just turned 100 December 24th on Christmas Eve, and she's in a rest home. And so the last time I saw her, I kid you not, I was like, we were going through the family album. That's your son. That's your daughter. Wow. No, son, daughter. Yeah, you know these people. Right? What's the quality of life when you get up to such age? Yeah. I often wonder if we lived to 900 years or something in such a decayed state, would it be worth it? But then again, is that necessarily how people lived? If Adam lived to 930 years? There's a lot of suggestions that the world before the flood was very different, right? So there was this thing about the quality of life. What would it be like? There was an interesting article I bumped into recently that actually our brains are not even developed when we're 20. Through our 20s, our brains are still wired. So there's a lot of suggestions that there's much more going on. There's all this machinery. There's all this equipment we have to live much longer. So it often makes me wonder, is like, are we even mature when we think we're mature? Right? Do we still have so much to learn, so much to understand? So I don't know if I put the verse in here. So, oh yeah, so the quote. But so, I just want to say, so there's that bit when God says, I should have put this verse in here, when it talks about, I will not put up with man forever, his years would be 120. That's about all I can take. It might have been specifically for me, I don't know. Shawn, that's yours, it's too much. Running amok, playing video games. So of course that was another thing that made me stop and go, how interesting. So I just tried to show you a slightly different perspective of some things that have made me go, right? And part of this, part of the reason why I put these up here, so I have people seeing these, right? So you look at them and you suddenly see something different, right? It's a rabbit. No, it's not a rabbit, it's a duck. It's a duck. Or it's like, It's an elderly woman? No, it's a young woman. She's looking that way. So it's a saxophone player. No, it's just a woman. Right? Sometimes it takes a while for you to see the different perspective. But so even from like a scientific perspective, you can bring preconceptions, right? Innocent, unbiased observation is a myth. There's always preconceptions when you go and you look at data. There can be no observations without an immense apparatus of pre-existing theory, right? So a lot of scientists are trained to see things from a very specific view. And so I, as a Christian, over the years, have just seen some of these things. It was like, well, wasn't that interesting? So of course, we have 7-day rhythms for healing and recovery. What was the word? What was the word? Circumception. That was cute, guys. Circumception. So what was the other one? Biological kinds from biological data. So that was the typological thing. I'm sorry? Yeah, typological view. We have a mitochondrial mum. I couldn't think of another. It's mum, right? Mum. The suggestion that there was a massive population decrease, right? So it looks like the human race went through a sieve of some kind. And immortality, we have immortality inside us. And so what's the word for that one? What do you think the word that one's going to be?
Mortality?
Mortality, well, I was thinking, how about, it starts with a T. I'll give you the T. Telomeres. Does that sound suitable? So telomeres. Okay, yeah. So that's basically it. And I was going to say, we do have a merchandise for sale, so if you want to fix your telomeres, it also helps headaches, neuralgia, cough, cold, sneezing, and also provides immortality for a nominal fee. Just please feel free to pick up your bottle on the way. Okay, that's pretty much what I got for today. So thank you.